students, यहाँ पे हमें आज okay. Good morning, dear students. Our topic for today's lecture is fertilization in angiospermic plant. So first we will discuss about the term fertilization and angiospermic plant. So fertilization is what is the process of fusion in angiospermic plants. So angiosperms are the highest category of plants, highest plant category which have the complex development system, complex organ system in plant kingdom. So first, why it is necessary? What is the need of fertilization in plants? So fertilization first of all, it is a process of fusion of two gametes, two haploid gametes, and it generates diploid gametes. So first important thing is that it continues the progeny, continues the life cycle of a plant. Like in human beings, we reproduce babies. So in plants also there is production of embryo that produces seed, and that continues the life cycle of another plant. So first thing is it is essential to continue the life cycle of plant. Then next, it is essential because it develops endosperm and zygote. Two things are developed during the process of fertilization. First is endosperm and the second is zygote. So zygote continues the life cycle of a plant, and endosperm provides the nutrition to the developing embryos. Such in the case of humans, as the womb of the mother provides the nourishment to the baby, same in the case of plants, there is a tissue known as endosperm which provides nourishment to the developing embryo, which further generates seed. And the next thing is this process utilize the male gametes, that is pollen grains. If they are not utilized, they will become waste, because that is a male gamete and that is used during the process of fertilization. If we do not use them, they only cause allergy, so they will totally waste, and they create genetic variation because different type of fusions take place. So variation occurs through the process of fertilization. It is essential to maintain life cycle. And the last thing is it restores the number of chromosome, because haploid fuses with haploids and it generates diploid that becomes double in number. So there is a restoration of number of chromosomes. So these are the things which are essential, which occurs during the process of fertilization, and these are the importance of fertilization. Suppose if you have to tell that why fertilization is essential, then you can tell these things. Now we will discuss about the general structure of a flower. So what is a flower? Flower is a reproductive organ of a plant. Because we do not know, if we know we want to define flower, then how can we define that flower is a reproductive organ of any plant. So here we will discuss general structure of a flower. This is the general structure of a flower. Flowers are of two types, complete and incomplete. So complete means there are four whorls in a flower. That is, first is calyx, that is known as sepal. The other is corolla, that is known as petal, and then stamen, and then carpel, that is female. So there are four whorls, 1, 2, 3 and 4. So if these four are present in a flower, then we call that flower as a complete flower. And if any of them is absent, suppose if 3 are present, if 2 are present, if nil is present, because stamen and sepal and petal are always present in a flower. So if male or female are absent, or female is absent only, or male is absent only, then that flower is known as incomplete flower. So here we are discussing about the complete flower. So first, whorl means from outside to inside. So first whorl is sepal. The single member is known as sepal, and the whole group is known as calyx. So calyx is the first whorl and its single member is sepal, and it is always green in color. It is attached with the stem of the plant. Then next is petal, that is known as corolla. Group is known as corolla and single member is petal. It has different colors like pink, red, white, purple, that depends upon the pigment available. Then third is male, that is known as stamen. It has we will discuss separately about the structure of male, and it is divided into anther and filament, and this one in the central part is female, that is divided into stigma, style and ovary. So during the process of fertilization, these two organs are used, these two walls are used, that is male and female. So this is a general structure of flower. Flower is what? Reproductive organ of any plant, and it has four whorls. If it has four whorls, then it is known as complete flower, and if it has any there is absence of any one of them, then that is known as incomplete flower. So this is a flower, and this is a general process of fertilization. So we start from here. This is a plant, flower is attached with this plant, and during the process of fertilization, first of all, this is a female part and this is male part. So pollen enter, that is anther, pollens are dehisced from here, they enter inside the female and they fuse with the egg nucleus, they form the zygote that develops into embryo and that develops into seed, and when we sow the seed inside the soil, that develops into plant. So this is the process of fertilization. Fertilization is just the fusion of male and female, and after fertilization a structure develops that is known as zygote, and zygote forms the embryo and embryo forms the seed, and the seed is sown inside the soil and it germinates into plant. This is the complete life cycle of a plant. So we will describe in detail. This is a generalized diagram. So these are the steps which takes place during the life cycle of a plant. First is microsporogenesis, then microgametogenesis, pollination, megasporogenesis, megagametogenesis, fertilization, embryogeny and endosperm formation. So microsporogenesis and microgametogenesis, these are the stages during the development of male gametes, that is pollen grain, and megasporogenesis and megagametogenesis are the steps of the development of female gamete, that is known as ovule. And pollination is the transfer of pollen grains from the anthers to the surface of stigma. So first is the microsporogenesis. Sporogenesis is the formation of microspores. That microspores are what? Pollen grains that are formed from the microspore mother cell. We will discuss with the help of a diagram. I am just telling what are the steps and how can we define them in a single line. Microgametogenesis is the development of gametes from microspores, formation of male gametes from the microspores. Then pollination is transfer of pollen grains from anther to the stigma, and megasporogenesis is formation of megaspore formed from megaspore mother cell. And then megagametogenesis is the formation of embryo sac from megaspore. And then fertilization is a fusion of male and female gamete and formation of zygote. Embryogeny is the development of embryo from zygote. And last is endosperm, formation and development of endosperm as a result of triple fusion or double fertilization. So these are the general steps which take place, and then the process of reproduction completes. So we will discuss first about the male gametes, and then about the female gametes, and then about the process of fertilization. So this is the general structure of a male. So this is anther, this is filament, and this is the connective which connects anther with the filament. And when this anther is dehisced, when we do transverse section, we cut it from the TS, then we will see there are four chambers. Let's discuss about its structure also. Stamen is what? Is a male part of a flower. It consists two parts, that is anther and filament, and connective is a link which connects filament with the anther, and proximal end, that is nearest end, is attached with the thalamus. Thalamus is what? Thalamus is the base of a flower. When we discussed about the structure of the flower, the base where the petals start to form, sepals start to form, then that is the thalamus. So that proximal end is attached with the thalamus, and the number and length of stamens vary differently. Matlab means they are different in different flowers, so we cannot say that there is a fixed length or there is fixed number of stamens. They vary in all the flowers, in all the plants. So let's discuss about the internal structure of anther. Anther is bilobed structure. So when we cut the transverse section, it looks like this. There are 4 chambers, and there are 4 layers also. The anther is surrounded by 4 layers, and first is epidermis, endothecium, and then last is tapetum. And pollen grains are situated inside the pollen sac, and there is a thin layer that is known as stomium, that is also present inside the. This is a young anther, and this is a mature anther. This is a TS section, like transverse section. When we cut from the middle, a normal dithecous anther is made up of 2 anther lobes, and when we cut, it contains 4 chambers, and pollen grains are situated inside the pollen sac. So this is the description of that diagram. There is a wall and pollen chamber. 2 structures are there. Wall is divided into 4 layers, but first is epidermis, that is the outermost and thickest layer. Endothecium is next to epidermis, and this is a single layer which helps in the dehiscence. When the anther pollen sac bursts, then the pollen grains are dehisced with the help of this layer. Middle layer is 3 to 4 layers, thin walled structure situated just below the endothecium, and it is disintegrated at the time of anther matured. When the anther matures, this layer disintegrates, means we will not be able to see this layer. We will able to see this layer during the anther is young. And the last is tapetum, that is the innermost layer, that is multinucleate, densely cytoplasmic, and it provides the nourishment to the developing microspore. It is of 2 types, secretory or amoeboid. And the next thing is pollen chamber. Pollen chamber is a cavity of anther lobe where the microsporogenesis takes place. Microspores develop from inside the pollen chamber. So how development of anther or how development of pollen grain takes place, and then this is the general outline diagram of that. First of all, there is a tissue, that is parenchymatous tissue and epidermis. So one of the cell of these tissue is highly dense cytoplasmic with dense nucleus and is large in size, that is known as archisporial initial. There is not a single archisporial, there are many. So we need to identify that. There is first mitosis, and the archisporial cell divides into 2. First is primary parietal, and then sporogenous cell. Primary parietal cells develops the layers of anther, and the sporogenous tissue develops in the inside lobe of the anther. So this through these steps the development of anther takes place. So these are the 4 layers, and this is the sporogenous tissue, or this is spore mother cell, which develops the microspore. So this is the general structure of development of anther. Next we will discuss development of male gametophyte, that is pollen grain. This is the flower, these are anther. Microsporocyte develops through meiosis into 4 microspore, and these 4 are functional, and each of the 4 microscope generate 2 nucleuses, that is generative nucleus and tube nucleus. And the tube nucleus and generative tube nucleus remains inside the tube, and the generative nucleus fuses with the egg cell. This is the general process. Now this is the these are the stages of gametogenesis. We have studied the microsporogenesis, the development of pollen grain. Then this is development of microspore mother cell and pollen grain. Then the pollen through the pollen grain we will discuss about the development of microgametogenesis. This is a pollen. It has 2 layers, exine and intine, and there are 2 nucleuses, that is tube nucleus and generative nucleus. Both are functional. This is the development of pollen tube, generative cells and tube nucleus. Tube nucleus is vegetative and generative means which takes part in the process of generation, that is the fusion of gametes. So this tube nucleus remains inside the tube, that does not take part inside the during the process of fertilization, during the process of fusion. And the other nucleus, generative cell, divides into two through the mitosis, and it generates two male gametes, and these two take part in the process of fertilization. And this is the final pollen tube, male gametes, two generative and one tube nucleus, and this is callus plug. So what is callus plug? This is another question here. Callus plug is what? It is made up of beta glucan, and it restricts the entry of pollen tube back inside the pollen tube, means they cause the pollen nucleus to remain inside this area only, to remain in a small area only, so that they do not go back to the pollen tube and they move through the stigma inside the ovary. So this plug helps them. Plug is means which block that particular area, that do not allow the things to move backward direction. So that is a small cytoplasmic area is created by callus plug. So this is very important. If this plug is not there, they can also move back to the pollen tube. So this is microsporogenesis done and microgametogenesis done. The next thing is pollination. We will not discuss that because that is another separate topic. Now we will discuss about megasporogenesis and megagametogenesis. Megasporogenesis is the same like the microsporogenesis. First, archisporal initial, any cell which is dense in cytoplasm and which is conspicuous, which has conspicuous nucleus, which has large size, and it has all the organelles well developed, that is known as archisporal initial. That divides into two, and then primary parietal and sporogenous cell. Primary parietal tissue develops the nucellus region, and the sporogenous tissues develops the megaspore mother cell, and the megaspore mother cell divides through mitosis into 4, and out of 4, 3 degenerate and 1 remains the functional megaspore. This is megaspore mother cell, this is nucellus region. Here we can see this is megaspore mother cell developing megaspores, and nucellus developing. The nucellus is the extra tissue which is present outside the megaspore mother cell, that provides the nutrition to the developing embryo, that transforms into nutritive tissue, and that is a thin layer. That, and the next thing is megagametogenesis. So megagametogenesis is what? Development of female gametophyte from megaspore mother cell. Here we know the 3 degenerate and the only 1 remain functional, that divides and form 8 nucleuses through the 3 consecutive mitosis. They form the 8 nucleuses first, then 4, then again 8, and they form the 8 nucleate cell which arranges like this pattern. So embryo sac is what? That is female gametophyte. Development of embryo sac from from megaspore mother cell is known as megagametogenesis. So on the basis of their development, there are 3 types of embryo sac, that is monosporic, bisporic, tetrasporic. That is depends on the number of megaspore taking part in the development of embryo sac. If only 1 takes, then monosporic. If 2, then bi, and if 3 takes part, then that is 4 takes part, then that is tetrasporic development. So only 1 remain functional, and it divides mitotically and then forms the 8 nucleate and 7 cell structure, that is known as embryo sac. So here are different types of cells. there are two ends. We will discuss separately about the structure of embryo sac. So there are two ends, that is micropyle and chalaza, and there are total 8 nucleus and 7 cells. So 3 cells are here, 3 cells are here, and one cell is here. So number of cell is 7 and the number of nucleus is 8. So this is the structure, that is 7 celled and 8 nucleate embryo sac. This is a type of polygonal embryo sac. Monosporic embryo sac means only one megaspore taking parts in the development of embryo sac. So here are 8 nucleus, each cell has single nucleus, only this cell has 2 nucleuses. So here the development of nucleus, when a fusion occurs, there becomes 3 nucleus, so this becomes 3n in number, and the others remain only 2n, others remain only 1n. Synergids are arranged and egg is arranged at the micropyle end, and antipodals are arranged at the chalazal end, and there are two ends inside an embryo sac. These are synergids, and there are another structure that is known as filiform apparatus. Filiform apparatus is a thread-like structure made of parenchymatous tissue that provides the direction to the entering pollen tube, that provides the direction inside the ovary to the entering pollen tube, that where it has to go and where it has to fuse with the egg. So egg cell is present between the synergids, and synergids contain filiform apparatus that attract and guide the pollen tube, that directs the pollen tube towards the ovary. This is the embryo sac we has discussed just now. Chalaza end, micropyle end, integuments, and this is nucellus single layer. We discussed that parietal cell form the nucellus. This is 2 to 3 layered or single layered, and this is the inside the ovule. So this is the stalk of the ovule, that is known as funicle, and that is ref. So this is the structure of LS of ovule, like LS is longitudinal section. So during the development of female gametophyte, first is megasporogenesis, that is development of megaspore from archisporal initial or megaspore mother cell, that is one surviving megaspore, megasporangia, megasporocyte, integuments and micropyle, and this is the development of, this is megagametogenesis, that is development of embryo sac from megaspores. 3 antipodal cells, 2 polar nuclei, 1 egg cell and 2 synergy. So this is the structure. This is known as embryo sac or female gametophyte, which has 7 cells and 8 nucleuses. 2 nucleuses are arranged in a center, that are known as polar nuclei, that are 2 in number, and other than that all the cells are single in number. So now we are done with sporogenesis, megagametogenesis, microsporogenesis, microgametogenesis. We have male gametes also, we have female gametes also. Now the next step is what? Fusion of these gametes. So fusion takes place. This is the general structure we have already discussed. This is generation of male gametes, this is generation of female gametes. Now there comes the process of fertilization. Fertilization starts from the stigma. This is the general structure of a female, and it contains 3 parts. First part is known as stigma, and the other part is known as style. This whole long tube like structure, and this part is known as ovary. So it has 3 parts: stigma, style and ovary. Ovary is this whole part, that is lower most part of a female, that is known as carpel, and stigma is the uppermost, that is works like a receptive for the pollen grains. Pollen grains come at the surface of stigma, then they form pollen tube, and the pollen tube enters inside the style. So here we can say this yellow structure is pollen tube, and it contains 3 nucleus. 2 are generative and 1 is tube nucleus, and that will enter inside this section that is known as ovule, and it contains egg and it contains pollen nuclei also. So we have to fuse that male gamete, that is pollen, with the male nucleus or pollen nucleus with the female, that is egg nucleus. So this is from there the process of fertilization start. This pollen tube enters inside the ovary through the style, and filiform apparatus provides the guidance direction to the entering pollen tube. So entry of pollen tube can be of 3 types depending on from where it is entering inside the ovule. First is progamy, then second is chalezogamy, and third is mesogamy. Progamy is what? The entering of pollen tube through the mouth, through the micropyle end of the ovule is known as entry progamy. And second is chalezogamy, as we can see through the name chaleza. If the pollen tube is entering through the chalazal end, that is known as chalezogamy. If the pollen tube is entering through the integuments, then this is known as mesogamy. So that is different in different flowers. That depends on how pollen tube is entering, from which mode it is entering. That is on the basis of that it is divided into 3 types. If the pollen grain germinates, pollen tube grows, and pollen tube moves inside the ovule, inside the style, and now the process of fertilization takes place. Pollen nuclei, egg, synergids, and 2 sperms have entered. So out of these 2, the pollen tube discharges its sperm inside the female gametophyte. One sperm out of that fuses with the egg cell, and one fuses with the polar nuclei. So polar nuclei, if one fuses with one fuses with the egg cell plus 1 polar nuclei, so it forms 2n, n plus n, that it forms 2n, that is known as zygote. That develops into embryo, and this polar nuclei, that is already 2n, plus 1 sperm nuclei fuses and it becomes 3n, and it develops into endosperm. So polar nuclei will develop into endosperm nucellus, that is 3n, and egg cell will develop into 2n, that is zygote. This process is known as double fertilization or triple fusion. Why? Triple fusion means fusion of 3. Here we can see that it becomes 3n. It becomes 3n. How? 2n plus n is equal to 3n. So this is known as triple fusion, because there is fusion of 3 nucleus. There is fusion of 3 nucleus, and it is known as double fertilization because 2 types of fertilization are taking place, one with the polar nuclei and one with the egg cell. Therefore it is also known as double fertilization. So what is the significance of double fertilization? It restores the diploid chromosome number, and again it forms the endosperm, which is very essential for the nutrition of developing embryo, and embryo is developed from the zygote. Okay, so these are the factors which can affect the process of fertilization. Pollination is very important because if pollen grains do not come on the surface of stigma, then fertilization will not take place, and there are many modes of pollen grains, many modes through which pollen grains come to the surface of reach to the surface of stigma. That is through the wind, through the water, through the insects, through the birds. Through the wind, that is known as anemophily. Through the water, that is known as hydrophily. Through the insects, through the birds, or through the animals also, they can also reach to the surface of stigma. Male sterility. If the male is sterile, means it is not fertile, then if the pollen nuclei reach to the surface of female, then also fertilization will not take place because the male is already sterile. Self incompatibility. Compatibility is what? Male and female should both be compatible with each other, means their chromosome, their fusion, there should not be any hindrance between the fusion of these two. If it is incompatible, that flower is incompatible, then fusion will not take place. Viability of pollen grains. Viability means growth, uh, life of the pollen grain. If the pollen grains are they have good life, they are viable, then the fertilization will process will take place easily. If the viability is not good, because viability is also tested if we want to do artificial fertilization, if we want to do fertilization lab condition, then before making fusion of the male and female, we will have to check the viability of the pollen grain, so that we will know that if fusion will take place or not. Then next is availability of nutrition. For the growing pollen tube, there should be proper nutrition available. And receptivity of stigma. Stigma is how much receptive. There are some chemicals which secrete on the surface of stigma that attract the pollens, and they attach cohes the pollen on that surface, which helps in the entry of pollen tube. So this topic completes here. We have done with the fertilization is what? It is the process of fusion of male gamete with the female gamete. So what is male gamete in plant? That is pollen grain and pollen nuclei. Pollen grain is actually a microspore, and male gametes are generated through the pollen grain. It divides, its nucleus divides, and then it generates pollen nuclei. There are two in number, and generative and vegetative. And the female development of megagametogenesis and megasporogenesis is the development of female gamete, that is known as embryo sac, and inside the embryo sac development of fertilization, process of fertilization takes place. Pollen tube enters through the stigma, and it releases its nucleus inside the two nucleus inside the female body, female gametophyte, and then a fusion of two nucleus takes place. One fuses with the pollen and the one fuses with the egg. It forms a zygote and endosperm, and this process is known as double fertilization, because two fertilization are taking places. And this is also known as triple fusion, because there are fusion of three nuclei in the pollen nuclei with the sperm nucleus. So this is also known as triple fusion. And why it is important? To continue the life cycle, to maintain the number of chromosomes, and to generate the variation of genetic diversity, and that is all. Okay, and these are the factors which affect the process of fertilization. These are the important points which you should learn, like which part fuses with which and what it forms. Egg fuses with sperm, and that forms a zygote, which develops into embryo on maturity. Then pollen nuclei fuses with sperm, which develop into endosperm on maturity. Integuments develop into seed coat, coating of seed, like the layer of seed, the peel of seed. Nucellus develop into perisperm. Micropyle develop into mouth of the seed. Funicles develop into hilum. Ovary wall develops into fruit. Ovary develops into fruit, and ovule develops into seed. Fine. So these are the structure which on maturity they develop into these structures. As we can see, seed, there is a peel, that is known as pericarp, and seed coat is known as testa, or we can also say seed coat. So this is all for this topic. Thank you.